Now let's start with the workshop. I will lead you through this objective and the refractive index workshop. I will give you some introduction slides, and then later on we will do some quiz or exercise to work on with the data that I acquired of the matched and the mismatched point spread function with the different objectives. So actually, here's the potential learning outcome of this workshop. We will, in this work, we will do the following. First, I will show you that we will not really acquire. I will show you how we acquire the image of the sub-resolution fluorescence beads with a single point confocal to get the point spread function width, and then explore the data with different combinations, because we will have the point spread function of the matched refractive index and the mismatched refractive index. We will compare the data that and do a small exercise. And I will also use 3 types of sample and 3 objectives to demonstrate what happens to the image if your sample match the refractive index or do not match. See the difference of these combinations. Okay, first, actually, this will already have been shown to you by Sylvie. I will not go through these slides again. I hope you already understand most of them. What I want to mention here is that the point spread function is one of the key parameters that for us to access how well a microscope performs, or how well a microscope can distinguish 2 very close dots. It's called the resolution of the microscope. And actually, the sample we are going to use, or we are going to measure for the point spread function, is like this. We will have a cover slip and also a slide. The distance between them is 50 microns. And on the surface of the cover slip, we will coat with 1 layer of the fluorescent beads. This fluorescent beads is very small. You see the diameter is only 100 nanometer, which is a small one, because we need to have this very small, because this need to be smaller than the theoretical X and Y resolution of the microscope, which usually is around 200. And the color here we chose is red, but we can also choose other color, depends on what we want to measure. But here we only choose 1 color, which has excitation of the 580 and emission around the 605 nanometer. And this beads, another layer will be on top of on the top of the glass slide. Actually, it's just which means it's 50 microns away from the cover slip. And in between, we will fill with either air, water, or oil to mimic the different samples. And for the objectives, we will also use different objectives. We will also use air, water, or oil objective to measure the point spread function both on the cover slip and on the glass slide, which means the 50 microns away from the cover slip. So in total, we will have 9 different combinations, and for each of these combinations, we will have the point spread function in both on the cover slip and also on the glass slide. This is just to simulate different types of thickness 6 samples, which has different refractive index imaged with different types of objectives, just to show you the effect of the different matched or mismatched combination of the objective and your sample. Here is the microscope we will use. Here I chose the single point confocal that works quite well in our facility. But in theory, we can also use wide field or spinning disk to measure the point spread function. And here is the image of the sample that I used to acquire the point spread function. You see it's a glass slide, and down below is the cover slip. In between is 50 microns distance, and below is our objective. And here's the overview of the sample. Again, actually here, because it's an image, we cannot see the cover slip or the glass slide as they are transparent. But actually, you can see one layer of the fluorescent beads on the cover slip and another layer of the beads on the glass slide. In between, the distance is 50 microns. And this image or this 3D reconstruction was captured with the air sample and with a 20x objective. This is just to give you an overview of this sample that we used. And for the real measurement of the point spread function, we will use the 60x objective with air, water, or oil. You can clearly see that we have a lot of beads on each of the layer, and maybe one bead you notice is this one, this very bright one. This very good, has very good contrast of the compared to other samples. Is it possible for us to use this one? If we zoom in, we can see actually this is how it looks in the 3D environment. It is much larger than the other beads. Anybody knows why is this? Actually, this is because this is a cluster of the beads. It's several beads formed aggregates. That's why it's larger and much brighter. But as I mentioned, we should use the beads that is sub resolution, which means less than 200 microns. So we should avoid it. We cannot use these beads because we know it's a cluster. It's much larger than the other beads. And what we should use or to measure is single bead without neighbors. For example, all these ones we could not use them either because they have neighbors very near. It is difficult to distinguish if we zoom in. What we should use is something like this. They are bright, single, and there's no neighbors. And then we can then choose any of them, then switch to 60x objective. We can, for example, here is the oil objective, but in the different combination, we might use either air or water or oil objective. And also for the sample, which means the gap between the cover slip and the cover slip, it can be filled with either air or water or also oil. And what we are going to do next is to localize to this single dot, and then do a Z stack. Find the bottom of the Z position and the top of the Z position. And the start and end plane should be far enough because we want to capture the whole signal from the beads. If it's not enough, we might lose some of the information. Here is it. This is just some background noise because we still haven't reached the beads yet. But here you can see gradually, when scan gradually to the top, we are reaching and then leaving the beads. And this is the signal from the single fluorescent beads, and the whole stack. Based on the whole stack, we will be able to get the point spread function of the single beads. And here is the tile of the previous images. We can see that in total, we scanned 251 Z focal planes, but only in the middle part of the focal plane, we have the signal, because actually this is because we have the, here we have the oil objective with oil sample, which means that we have the matched refractive index. So we have relatively small point spread function. I will show you soon how the mismatched point spread function looks like. And here, this is a 3D reconstruction of the same point spread function, or your sample, or your objective. You see how it looks like. Just to remember, actually, this is a signal from a single regular fluorescent sphere, which originally looks like this. But in reality, when we image from the microscope, it's elongated already, even we have the matched refractive index. This is because that in that we have this. I think Sylvie just explained to you why this is. Just here is just the example of to show you in real life how it looks like. Okay, just now we are seeing this or your objective or your sample, which has matched the refractive index. And here I will also compare with you another combination, still all your objective, but here we will have some air sample, which means that we have mismatched the refractive index. Here is how it looks like. We also, it's the same scan range. We still, you see, very early, we already can see the signal from the mismatched sample. And actually, it lasts almost until the very end of our Z scan steps, which means that if we look at in the 3D reconstruction, that because this is a sample actually, when we reconstruct, you can see in that direction, it's very, very elongated because we have the mismatched refractive index. But if we have the matched refractive index, no matter whether it's on the cover slip or it's on the slide, which means 50 microns away from the cover slip, we still have relatively small and very similar refractive point spread function with the cover slip. But if it's mismatched, the effective is the result will be huge, different very largely. And the next step after acquiring, or we assume that we have obtained all these different combinations of the matched and mismatched refractive index samples. What next step is that we want to quantify them. So we showed you actually how to quantify them. We have different ways. In this workshop, the way we will use is not a relay, but also the four full-width half maximum. This is because we are going to not to do it manually. We are going to use this image plugin called Metrology. When we feed in our acquired point spread function image stacks, it will provide us it will automatically help us to measure and quantify the point spread function. This is an example of how we analyze the data. We just use Fiji or ImageJ to open this image stacks we acquired, and then start this Metrology, the point spread function report generator, fill in the very simple information. For example, we are using the confocal and the wavelength. Actually, for confocal, in theory, we should fill in the excitation, but to be in agreement with Sylvie's suggestion, here we also use emission. For simplification, it doesn't matter too much, and also because this is only to calculate the theoretical resolution, and also the NA of our objective, and the pinhole used, then click OK. A PDF report will be automatically generated based on this image. And you can see this is a different view. This is a view of this point spread function in different directions. And also it will give us the measured resolution in X, Y, and Z directions. Of course, there's also theoretical resolution, but we will not focus on this for this workshop. We only focus on this X, Y, and Z resolution. Actually, how does this software measure this X, Y, and Z resolution? Is that, for example, for the X direction, it just draw a line across the maximum intensity of this XY image, and then based on this image, they fit this with the function, and then the 4-way half maximum of this function will be the resolution in the X direction. Similarly, for the Y direction, draw a line in Y direction, and then fit and have the 4-way half maximum of this direction. And also for Z, it's a similar strategy. And actually, we are not going to do this this experiment, but we are going to look at the tables or the results that I have acquired. You can see this is the point spread function results, and we have the beads layer on the cover slip, and also on the slide, which means 50 microns away from the cover slip. And for example, if we focus on the cover slip, we have the different combinations of the objective and sample. And then, for example, here is the X, the resolution in the X direction for the air objective with air sample. We have the resolution of 372 nanometer. Air objective water sample is 361, and so on. Later on, we will do a small exercise based on this measurement. You will try to explore this table with some questions and compare the results of the point spread function with matched and mismatched data. Before we go into that, I will show you something else, which is that the mismatched refractive index does not only affect the point spread function, but also leads to the wrong Z distance measurement. Sometimes we need to measure the distance or measure the size or measure the depth of your sample. But if we have the mismatched refractive index, it will give us the wrong measurement result also. Here, it's still the same sample. It's still the two-layer beads on the cover slip on the glass slide. The distance we already know, which is 50 microns. But we will measure this with different combinations of the objective and the samples. How we measure it? Actually, this is very simple. We just still use the Z-scan. We move the objective from the bottom to the top. And when we move it, we will see one layer with the beads, and then move up, up, up. And then you see here's another layer of the beads. Then what we do is that we record that position shown on the microscope, because when we reach the first layer with beads, we know this Z coordinates is this 2186, and when we reach the other layer of the beads, we know that the coordinates on the microscope is 2206 micron. We just minus of these two, then we get the relative distance measured by the microscope. This is just how we do it. I will not show you more than that, but just to show you the results that with different combinations of the objective and sample, Actually, if we have the matched objective and the sample of the refractive index, we will have very similar distance, which is very close to 50 microns, which we know this is a ground truth. We know that this because we know our sample. I made this sample to be that this distance of this sample is 50 microns. But if we have a mismatched refractive index, for example, if you have an air objective, but the sample is water or oil, we have the measured distance is smaller than the actual one. While vice versa, if we have the oil, for example, use oil objective to measure the air or water sample, then the measured distance would be much larger than the real distance of the sample. Why is that? Actually, this has been explained a little bit by Sylvie in her previous lecture. Here, I will show you again. For example, for the mismatched objective and the sample refractive index, for example, here we have the air objective and a water sample, which means that the n of the objective is smaller than the n of sample. According to the Snell's law, this means that the theta here of the objective will be larger than the theta here of the sample, which means that the light going into the sample will be banded further away. But what we assume is that we always assume that light goes straight. We still think our sample is here, but actually we already seeing some place already far away. Actually, this is we think we measured. Actually, this should be the real distance, but actually this is the real distance. But what we measure, we think we're still measuring here, so the measured distance would be smaller than the real distance of the sample. In the opposite condition, if we have a oil objective and still the water sample, which means that the n of the objective is larger than the n of the sample. Still, according to the Snell's law, the light will be banded in the other direction. But we still assume that the light goes straight, which means the measured distance will be larger than the real distance that we have. Only when we have the matched refractive index of the objective and the sample, for example, we have both them with water, then which means that if the n is the same, then the angle is also the same. So the measured distance will be the real distance. So this is very important when you want to measure the distance. You should have the matched refractive index if you want to measure the distance of your sample further away from the cover slip. Here is the result. For example, if I have a matched refractive index and the sample, we should have the proper image. And if the refractive index of the objective is larger than the sample, we will have elongated measurements. Actually, we will stretch the sample because we thought we are measuring something further away, but actually it's not. On the contrast, if the objective of the refractive index is smaller than the refractive index of the sample, then the measured distance will be smaller than the real distance, which means that we will compress our samples in the measured results. Okay, this is it. And what we are going to do is that we are going to do a small exercise. And I can show you. You can go to this course module. There's a quiz called Objective and Refractive Index Mismatch. And you can open it. And you will do this in groups. All the groups will answer the question number 1. And then group 2 answer question 2. Group 3 answer question 3. Group 4 answer question 4. And group 1 will answer question 5. And I think we should have around maybe 20 minutes. If any of the group already finished the question, for example, assigned to your group, you can also try to answer other questions assigned for other groups. And we will come back and I will give you a summary of all the questions in around 20 or 30 minutes. Okay, I will stop sharing. Let me see, I can create 4 breakout rooms so that you can go to open.